basic science is essential, I would say, for improving therapies for patients. And we have been very pleased that we have this connection now between the European Association of Cancer Research and ESMO, which actually joined forces because both realized that we need each other. And so this is the first meeting where we really together put a program together and where we try to induce the interaction between basic researchers and clinicians. I selected 2 abstracts that were sent in by young investigators because I think they sort of a reflection of the interest of the next generation in showing what they are doing in the field. The first abstract relates to the sequencing of cell-free DNA in the serum of patients, a work presented by Sumanta Pal during the session.
What we've reported here at ESMO this year is one of the largest cohorts of patients assessed by cell-free DNA to date. In fact, the size of our cohort, more than 30,000 patients, actually exceeds many of the tissue-based data sets that are out there currently. The real question we're trying to address is the viability of this approach, and what we were able to ascertain is that about 84% of patients had detectable cell-free DNA in their blood. If you look across the spectrum of patients that we assessed in this 30,000 plus patient cohort, about 36% had lung cancer. There was also healthy representation of colorectal cancer, prostate cancer, and breast cancer. But we also had a spectrum of other rare cancer types, such as GIST, for instance. And I think that really speaks to the capability of this test to identify mutations with potentially actionable alterations. In this particular study with over 30,000 patients, what we've identified is the presence of multiple actionable alterations. Again, we were able to detect alterations in 84% of patients or thereabout. One of the other things that I'll point out too is that we were able to look at clinical outcomes in a subset of patients. This is primarily patients with lung cancer. And what was quite impressive is that amongst patients that had treatment linked to a specific alteration, response rates ranged from 50 to 100%, and disease control rates were nearly 100% across the spectrum of patients that we looked at. And this really speaks to the viability of using cell-free DNA as an assay to identify relevant therapies for individual patients.
I think this is a very important development. I mean, it's being used already by many labs, but this was the first time that they showed that you can do that routinely on a very large number of samples, and that you can, in this way, get a good idea of what are the aberrations in individual patients, but also to see how that might change during treatment. It's a very interesting study that shows a new component that might be important for the metastases of prostate cancer.
In this paper, we're looking at 2 questions, from the clinical point of view and from the biology point of view, because there's a lot of papers about CDCP1 published. Some showed that CDCP1 is anti-cancer, while the majority of the paper showed it's pro-cancer. So to verify these questions, we developed several transgenic models, including fly and mouse, to show that actually CDCP1 is an important oncogene. And the second part of the study was to check the expression of CDCP1 in human prostate tissues. And we screened for more than 400 human tissues, and we found that CDCP1 is upregulated in human prostate cancer, and this has a correlation also with the cancer aggressiveness. So the more the cancer is aggressive, the more you have CDCP1 expression.
They have shown that if you overexpress this particular receptor in a mouse model for prostate cancer, that it leads to a fast acceleration of tumorigenesis. And so the hope is that if we use drugs against this receptor, that we might inhibit this progression. And they showed evidence that that's certainly true in vitro. They have an antibody that can inhibit this protein, and there is a good perspective that this might also happen in patients. So it's a very promising strategy.